Morning, everyone. Thank you so much for coming out for our International Eczema Council meeting. I'm Dr. Larry Eichenfield from San Diego, and doing the honors of giving you a little background on the IEC and the program today, right at the bright start of the SID. Please scan this code to access program guide and agenda if you'd like. And so today's session's on what's new in pathogenesis and mechanism, drivers of disease. It's a great program that we set up this morning. And yeah, that's still me from UCSD, San Diego, where I'm doing the honors. I'm actually serving as president-elect of the IEC. And Alan Irvine just took over as president of the International Eczema Council. So this should be a great session, but as is typical of the IEC sessions, it is being recorded. And so I want you to know that you can access this session and other sessions in the future through the IEC website, so others who aren't here can have access to it as well. If you have questions or comments, please email info@eczemacouncil.org. And if people are interested about the IEC, potentially joining it as a counselor or associate counselor, that's the email address. We can contact Alan Stiles, who's in the back. He can wave his hand there, who works hard to keep the organization running. So background on the IEC, International Eczema Council is a global organization composed of 133 counselors and associates who we hope are leading voices in the world of atopic dermatitis, helping to move the field forward. Counselors are to be well-established, experienced clinicians or researchers in atopic dermatitis. And we have criteria where people essentially have to be leaders who have contributed to the field, both in terms of their published manuscripts, potentially in clinical trials, or leadership and scholarship activities. And the associate positions are set up for people who are rising stars in atopic dermatitis, who are moving to add information on atopic dermatitis and are establishing their careers. They have to have completed residency training and published at least 2 peer-reviewed articles and made meaningful contributions to the field. Now, depending on how you like to read data, I'll give you 2 little representations of the regions that we represent. We work hard to try to increase our international counselors and associate counselor distribution. You can see the 32% of our counselors are and associates are from North America, and then and Europe, which has been predominant, increasing number in Asia Pacific, South America, Africa, and the Middle East. And if you want to drill down of the countries that are involved, this is here as well. So this makes our regular meetings pretty difficult because we have wide time zone variations. So sometimes things are scheduled at 3 a.m. depending on where you are. But the idea, of course, is that atopic dermatitis is a global disease, and we have our influencers working together to advance the field from around the world. The IEC mission is to improve patient care by promoting research, education, and best practices in atopic dermatitis worldwide. The vision is transforming the lives of people with atopic dermatitis. Our anthem is a global community of leaders in research, treatment, and prevention. And our values are patient-centered, innovative, evidence-based, globally inclusive, collaborative, and forward-looking. I'd like to thank our corporate sponsors, our President's Council being AbbVie Pfizer, Executive Council Lilly Leo, our Director's Council Amgen Arcutis, Dermavant Galderma, Kawara Kirin, Sanofi, and our counselors Anapsys Bio, Cara, and Insight. The support is crucial because we have multiple different programs, both sessions like this, which I'm happy to say looks like it's getting pretty filled up, as well as a training fellowship programs and supporting atopic dermatitis research and training in multiple different programs. So we have a set of symposia every year. Today's is the third down. We were at AAD and Radla with very successful programs. EACI meeting, we'll have a session on safety of JAK inhibitors, and that's got to be Joint International Eczema Council and Psoriasis Council. ESDR, we have a marvelous session set up, again, an overlap of psoriasis and eczema with IPC and IEC, and EADV, chronic hand eczema. Again, these will be available if you're at the meeting or post available online as well. So we work hard to have continued symposia, which really put together tremendous state-of-the-art information. Shout out for fellowships if you have nothing to do over the next 2 weeks, and June 1st is the deadline. We have a variety of fellowships that are set up. There's clinical and research on-site fellowships, meeting their international opportunities for people to come and work with individuals. So this is not necessarily U.S. focused in terms of either the mentoree or mentors. So the candidates can be from around the globe. One is set up as a 4-week on-site and then continued collaboration off-site, so someone can come for a month and then go home for 11 months. So it's sort of feasible for people to drop in and get their experiences. And then there's 1-year research fellowships as well, where people have more intense research opportunities with mentors. Applications are accepted until June 1st, and go to the eczemacouncil.org to get the information. We specifically thank AbbVie, Pfizer, Sanofi Genzyme Regeneron for the fellowship support. So IEC also regularly puts out manuscripts and does special projects, try to advance the field. These are ideas that come to us from counselors, associate counselors, and other people who just send in ideas. So if you have ideas for what might be good to have, either consensus statements or a collection of research, please let us know. These have been very important projects over the years that I think have really influenced atopic dermatitis care and standard of care around the world. So tomorrow, for IEC counselors and associate counselors and industry who's been invited, we will have a breakfast reception. So this will be 7 to 8:30 in the Comet Room on the 27th floor, so we'll get a nice view from up there. We'll have just a light networking breakfast. Then I'll do a quick update on IEC and what we've done and what we're doing. Aaron Drucker will then be giving a presentation on living network meta-analysis to compare systemic treatments, and then we'll have a little informal networking afterwards. So please join us tomorrow if you can. For people who are social media connected, please feel free to like us, connect, and follow with the platforms. It's a way that influence happens around the world, so please comment on posts. General questions, as I said, Alan Stiles who at eczemacouncil.org if you have questions about IEC. And let's turn over to what we're doing this morning. So our chairs this morning are Emmer Gutman, who's the Waldman Professor and System Chair of Dermatology and Immunology, Director of the Center of Excellence for Eczema and Occupational Derm Clinic, Director for the Laboratory of Skin Diseases at Mount Sinai, the Icahn School of Medicine, and Carolyn Jack, Assistant Professor at McGill, Lead of Quality and Innovation in Division of Dermatology and Department of Medicine, Experimental Medicine, Microbiology and Immunology, and the head of the MUHC Center of Excellence for Atopic Dermatitis at McGill. Also a scientist in the Center for Translational Biology, ID and Immunity and Global Health Program and Research Institute of the McGill University Health. So we have two fabulous people to lead us through this session. Emma will come up, and I will leave the podium. She'll take over and do the program overview, and then go on into her lecture on epithelial barrier and immune markers. We have Gil Josipovich to discuss neuroinflammation and AD. Alan, mechanisms and biomarkers of AD prevention, which is pre-recorded. He's not here from Ireland this morning. Then we'll have a nice break and come back for the afternoon with Pat Brunner on navigating big and/or metadata. Carolyn will then speak on IL-13 protein concentrates in the epidermis of lesional and non-lesional skin of AD patients. And these sessions now move to the highlight SID abstracts of particular interest to the group. Shirley Paraja from Wake Forest on using facial skin AI to capture eczema patients' response to therapy, and Austin Moon from UCSD, my institution, will be giving a very interesting paper on topical moisturizer impact on survival of Staph aureus and Staph omitis in atopic dermatitis microbiome. So at this point, Emma, I'm turning over the podium to you. Thank you very much.
Thank you, Larry. And since I think you already introduced the symposium, maybe we will start with my own lecture. So I was scratching my head, what should I be giving today? And I wanted to give some new data on what's shared and what's different in the immune and epithelial barrier mechanisms across atopic conditions. These are my disclosures, none actually relevant to today's talk. So atopic dermatitis, a disease known to all of us, and by the way, it's the most prevalent skin disease we have now. About 7% of the adults in the United States, around 15% of the children. We know for a while that it has two hypotheses that basically are not mutually exclusive, and it has a vast immune and barrier abnormalities. In terms of immune abnormalities, we have abnormalities in IL-4, IL-13, IL-31, IL-22, so type 2 and 22 abnormalities with some contribution of Th1 and Th17 immune pathways. And we know also that it has vast barrier abnormalities with abnormalities in the epidermal differentiation complex markers such as filaggrin, loricrin, and others, tight junctions, lipids, and others, allowing this penetration of irritants, allergens, and microbes, creating basically the atopic dermatitis phenotype. And recently, of course, we know there is this very important neuroinflammation component that you'll hear much more from Gil about, with an intractable itch that our patients have. So how did we get to this amazing success that we now have, right? Atopic dermatitis is the poster child of success in translational medicine. It wasn't such an easy path, but we started with bench studies of pathogenesis that had incorporated biomarkers, both skin and blood biomarkers. And these were very helpful to formulate pathogenic hypotheses, and we continue to clinical trials with targeted agents. And it's really important when these are targeted agents to really help us understand mechanisms of disease. And there were many, many cycles for one success story. So, for example, 9 failures and 1 successes, but like I tell my mentees all the time, not only successes, but even more importantly, failures are very important in life in general to help us frame the direction we are going to. So I'll focus some of my talk on a new approach, or newer, that you saw many publications from the last few years on, and these are tape strips. These are particularly important in atopic dermatitis, a disease that has so many children that are involved with the disease, but not only in atopic dermatitis, of course, many other diseases. And why is that? Because biopsies, while they are very, very helpful, of course, but they are painful, they are scarring, they are prone to infection, not feasible for longitudinal trials and also longitudinal studies, right? Clinical trials are longitudinal, sometimes quite long, so you cannot sample the skin longitudinally using biopsies. And of course, we need to remember the pediatric population, where definitely this is a very difficult task to do, quite impossible, frankly. And we utilize tape strips that my lab spearheaded perhaps a little bit different approach than others were doing. And we do both RNA-seq and proteomics on tape strips, and I'll show you some studies that we've done, not only in AD, actually, also in other indications. So first of all, we did a study with Robert Bissonnet a few years back. And here I'm showing you the profile of atopic dermatitis patients in lesional skin. It's a head-to-head study, biopsies and tape strips, just to understand if we can get the same phenotype in the same patients. And the answer is yes, there are some differences, but pay attention that the usual suspects are very well represented in tape strips. You see there IL-13 front and center, CCL17, CCL18, the new kids on the block, AX40, AX40 ligand are there, and many other markers. In biopsies, you see the same. Some are the same, like CCL18. Some have smaller expression. Remember, biopsies are diluted because the epidermis and dermis are together. But the hyperplasia markers, IL-22 and S100s, are definitely better in biopsies because of the location where the sampling is in tape strips. But surprisingly, or maybe not surprisingly, but to us it was surprising, itch and barrier markers are better represented in tape strips. You get better differentiation of these with much higher fold changes and better significance, and these include important markers of itch, including IL-31, our favorite one, but also TRPV1, 3, and others, and many barrier markers like filaggrin, filaggrin-2, tight junctions, and others. And this is a study that we published with Amy Paller, wanting to understand if we can capture well the phenotype of children with early onset atopic dermatitis within 6 months of disease initiation. These were babies very early on in their disease, and compare it to skin biopsies from the same children, and pay attention that the important markers that we know for atopic dermatitis, like IL-4 receptor, IL-13, CCL17, they are all there, and we got a very high correlation between our tape strip phenotyping and the phenotype in skin biopsies. This is a study that my lab was a part of it, a beautiful study published by Jacob Tyson, showing that tape strip biomarkers can also predict development of atopic dermatitis, and not only development of atopic dermatitis, of severe atopic dermatitis. So these are tape strips done at 2 months that were able to predict later development at 1 year and 2 years of development of AD. A really beautiful study in a pediatric cohort they were doing in Denmark. And we also asked the question, can tape strips also help us in either clinical trials or real life as therapeutic biomarkers? And the answer is yes. This is a study we've done in real life use of dupilumab. And we utilized tape strips to understand the phenotype or the changes in the phenotype of these patients before and after dupilumab. And what we see here, we see that the usual suspects are downregulated here with dupilumab, including CCL17 and many other immune markers. And you see also increases in barrier markers like filaggrin and loricrin in the study. Oh, sorry. We next, and this is not a head-to-head study because these are done separately, but we put them side by side. We wanted to compare these tape strip data with dupilumab in real life to our biomarker study in the clinical trial with dupilumab. And in that clinical trial, of course, we also had placebo. Here it's real life, so there is no placebo. But we put side by side different type 2 biomarkers such as CCL18, CCL17, CCL26, the best biomarker of dupilumab, and others. And I think you'll agree with me that the reductions in these are in the same line. So here I want to show you a newer study that is now in press in allergy. And this is a collaboration with Ioana Agache from Romania. She's a pediatric allergist and an asthma specialist. And she came to me a few years ago, liking our tape strip study, and she said, why don't we hypothesize that based on the skin, we can actually detect abnormalities that are in the lung of children with asthma that do not have atopic dermatitis. And you know, it's kind of a high-reward study, high-risk, high-reward study. I wasn't sure we'll find anything, but I said, okay, let's do it. And I think you'll agree with me that the results are really nice from this study. So why did we do this? I don't need to tell you how hard it is to scope children with asthma, right? This is the gold standard for biomarkers in asthma, lung biopsies and sputum, but it's really hard to obtain it in children. It's also hard to obtain it in adults, but definitely in children. So we definitely need minimally invasive biomarkers that will be surrogates for the lung disease. So we hypothesized that skin tape strips, and again, these are children that don't have any skin activity. They don't have atopic dermatitis. They only have moderate to severe asthma, allergic asthma. So we hypothesized that in these children, we will be able, through the skin, to provide a window to what happens in the lung. This is the study design, and as you see, we didn't have many children. This is a preliminary study. We had 12 children with moderate allergic asthma, 11 children with severe allergic asthma, and 9 controls. And I'll walk you through the study, and I think the results are very interesting of the study. So first of all, we found a very large number of differentially expressed genes, both in the moderate allergic asthma, but also in the severe allergic asthma. And what you can see here, when you look at the PCA, you can see clearly that moderate and severe asthma have the same disease, more or less, right? They cluster together, and very different than the healthy controls. And you can see the same thing when you look at the differentially expressed genes by the fact that severe and moderate allergic asthma have only one differentially expressed genes. But you see more differentially expressed genes. It's kind of like a continuum in the patients with severe allergic asthma compared to controls, as compared to moderate allergic asthma compared to controls. This is the demographics of the study. Pay attention that none of the children had concomitant AD. Of course, some of them had family history or a personal history of other atopic diseases. Remember, asthma happens later in life. Pay attention that the mean age is around 7. Remember, AD starts in the first year of life. Asthma starts usually around the time they are between 6 to 8, and this is exactly our cohort. And what we found was actually quite surprising to us. So pathway analysis in the skin showed a gradual decrease in markers of epithelial barrier, including epidermal differentiation complex and cornified envelope, lipids, and tight junctions between healthy controls to moderate allergic asthma and more in severe allergic asthma, but both moderate and severe were significant. And we found an increased Th1 immune axis, again, that was gradual between healthy control to severe allergic asthma. And we also looked at genes that are modulated by interferon gamma in keratinocytes. They had the same gradual increase between healthy controls to severe allergic asthma. Interestingly enough, we didn't find real trends when we look at type 2, type 17, or type 22, or maybe trends we found, but none of them were significant. Now, what we found, those of us that understand asthma, I became a little bit expert in asthma after the study, know that the most important pathways in asthma are pathways of aging and oxidative phosphorylation, and there are multiple publications about that. So we found that this regulation captured very well by tape strips, and again, it was in a dose-dependent, or not dose-dependent, it was in a severity-dependent manner, with much higher dysregulation in the severe allergic asthma, in mitochondrial respiration, and oxidative phosphorylation markers. We also found that markers of a lung extracellular milieu were downregulated in the skin. Again, much higher downregulation found in the severe allergic asthma, including in regulators of extracellular matrix and different collagens. And importantly, I always say that a biomarker, to be a biomarker, you need this to be correlated with severity of diseases, right? Here she took extensive severity measures, including early exacerbations, FEV1, that is more known to us, IOS, which is impulse oscillometry, and FeNO. They are all considered very good biomarkers of asthma. And pay attention to the very high correlations. These are really high correlations, right? Particularly in such a preliminary study, we found very, very high correlations with the severity of asthma, with some correlations being much, much higher than what was found actually in biopsies of children with asthma. And we found two genes that used together, they are able to predict with 100% accuracy patients with asthma from healthy controls with an AUC of 1, which is quite impressive. So 100% accuracy prediction of who has asthma and who is a healthy control. So to conclude on this part, a healthy skin from children with moderate to severe allergic asthma exhibits barrier dysregulation and type 1 skewing with no Th2 inflammation, at least in the skin. This is the skin. And metrics of disease severity, FEV1, FeNO, annualized exacerbation rate correlated very well with asthma biomarkers. And the lung-related gene classifier was able to discriminate 100% between asthma and healthy controls. And thus, the skin may act as a proxy for lung disease, both interacting with the environment and thus modulated similarly. Now, I want to move on to another collaboration I'm very proud of because it also led to a big NIH grant that now I'm a co-PI on. And this is a study with Kersey from University of Rochester. And she came to me with a very interesting proposal to do a preliminary unfunded study from her NIH birth cohort that led now to another grant. And her cohort includes old order Mennonites that she compared them to Rochester children. And I'll show you how we showed differences in the skin of these infants. So we all know that atopy in general, and particularly atopic dermatitis, starts in the first year of life. But the molecular features of skin barrier and immune development in neonates at risk for atopy are unknown. And this is at the base of the grant. And her original grant, Zoom-1, was a prospective birth cohort that included old order Mennonites and a Rochester infant. So the Mennonites are from a farming or a very rural area, about 65 miles from Rochester. They are very similar to the Amish population. And the Rochester infants grew in Rochester, so urban lifestyle. And it was designed to evaluate the effects of environmental exposure on immune system and barrier development. And evidence from that birth cohort suggested, and this is published, that early farm life exposure was able to reduce the risk of atopic conditions, including AD, food allergy, and other atopic conditions compared to Rochester infants. And some of the protective effects here included greater bathing frequency in the Rochester infants. So OM infants, for example, were bathing only like once a week, whereas the others were bathing once or twice a day. Higher rate of medication usage and vaccination in Rochester infants, and higher consumption of unpasteurized milk products by 18 months of age compared to Rochester children. And the aim of this study was to, and this is a very preliminary study, but we will do a much larger grant, to identify unique immune and barrier skin biomarkers that are able to modify perhaps the risk of development of AD and promote healthy skin using a minimally invasive tape strip approach. And to elucidate the differential skin barrier and immune profiles between these two cohorts of infants. So this is the study design. Her cohort had 76 OM infants, 74 Rochester infants, but this was a smaller pilot on only 7 OM infants, that none of them developed atopic dermatitis, and 7 Rochester infants that some of them developed atopic dermatitis and some of them did not develop atopic dermatitis. So we had 3 Rochester infants with AD and 4 without AD, and all the Mennonites did not develop atopic dermatitis. And this is the presentation of how many genes changed in each cohort. Pay attention to multiple genes being modulated in OM infants as compared to Rochester infants, and also in OM infants as compared to Rochester infants with atopic dermatitis. Quite a large number of genes. This is by RNA-seq. And we wanted to zoom in to understand what genes are the ones that were different between these groups. So we found that OM had increased expression in general of epidermal barrier markers, including key components of it: tight junctions, epidermal differentiation in lipids, cadherins, claudins, and keratins. These were highly expressed in OM and reduced in the Rochester children, regardless if they had or did not have AD. Also, OM showed upregulation of genes that are involved in protection against pathogens and also in barrier maintenance. Some of them are known to you, like IL-36G, IL-27, and others. And also among the top 27 downregulated genes in the OM kids, we saw an overall lower tone of atopic-related genes, as seen here, including those that are proposed to be pathogenic mediators of asthma and atopic dermatitis, and also a lower tone in general of type 2-related markers such as IL-13, IL-4 receptor, and also T-cell and dendritic cell migration and activation. And also other suspects that are important, such as CCL17 and CCR6 and others, such as TSLP receptor. They were also, again, lower in the OM children. I told you that bathing frequency was very different between the two groups, and we saw very, very high correlations between bathing frequency with select skin biomarkers, including those linked to asthma and type 2 markers, as you see here. So to conclude here, it's a small preliminary study, but we find the results quite exciting. And I think this contributes to the growing evidence that a farm environment impacts infant epidermal barrier and immune profiles, potentially protecting against atopy. And this pilot study, of course, shows the utility of doing a much larger scale comparison of cutaneous biomarkers, and we are doing it in a grant that Kersey is the corresponding PI, and we are 3 PIs in this study, so super exciting. And I'll move to my last subject, alopecia areata, that is a disease that I think now we start understanding that it starts to join the atopic march, or it joins the atopic march. And I think my group had some contribution in this. And it originated when we did the original studies with dupilumab. We started to notice that with dupilumab, some patients that had also alopecia areata, patchy alopecia areata for most of them, they started to grow their hair. And we were scratching our head because we didn't understand that phenomena. And why we didn't understand that phenomena? Because historically, alopecia areata, right, is a type 1 only inflammatory disease. And as scientists, I think we always need to keep an open mind. And the reason being is that atopic diseases, right, are type 2 inflammatory diseases, including AD, asthma, and others. But then you start looking in the literature, and And we know that now we do it in our patients, and it's quite accepted. Total IgE and also specific IgEs like dust mite and others are significantly increased in alopecia areata patients. And we also know that patients with alopecia areata show seasonal flares. And also, many of us know that antihistamines are promising as an adjunct to alopecia areata treatment, and now they are given together with many other treatments given to the patients. In addition to that, the largest cohorts show in alopecia areata show that the highest comorbidity is actually atopy in general and eczema in particular, and also population-based studies show increases of alopecia, increases of the incidence of alopecia areata in patients with atopy, and also the largest GWAS study done to date in the United States actually showed IL-13 as the highest association both in patients with atopy but also in patients without atopy. And from the DermPass literature, we see that cells associated with atopy are very prevalent in that infiltrate that is attacking the hair bulb, including eosinophils, but primarily mast cells. Majority of cases will have mast cells, and also a lot of the cases will have eosinophils as well. So that led my group to do a very large characterization in scalp tissues of patients with alopecia areata. And unlike other studies, we also compared to the two other poles of the immune system, psoriasis and AD. And we found that the Th1 pathway is actually shared to all of these diseases, maybe marking chronicity of the disease. The Th2 pathway was only seen in AD and alopecia areata, not in psoriasis. And IL-23, both P19 and P40 were also increased in all of these diseases, maybe higher differentiation seen in psoriasis. Also, we found that hair keratins are great biomarkers of alopecia areata. They are practically decreased or not there in lesional skin, not in non-lesional skin that is more like normal skin. But in lesional skin, you really have decreases of all the hair keratins, early, middle, and late hair keratins. And that all led to our dupilumab a study in adults, the original study. And in that original study, we found that patients with high IgE and or an atopic background, either personal or familial, they showed really nice hairy growth. It's not that the ones without these did not have any hair growth, but if you look at the graph, you see the best results in the population with atopy or high IgE. And we are doing another study only zooming in that population in adults, but I want to show you the molecular changes with dupilumab in the patients with AA and atopy and or high IgE. In this population at 48 weeks, we really reversed in tissues almost the entire transcriptome or profile, 97% reversal. And when you do by pathway, you look at 24 weeks, and at 24 weeks, only the type 2 is modulated significantly. Type 1 is not modulated at all. Only at 48 weeks, it's both type 2 and type 1. But hair keratins already are increasing at 24 weeks and more at 48 weeks, obviously. And what does it tell us? It tells us that probably type 2 is important early, whereas type 1 comes likely to play in chronic stage. And, you know, you don't need to believe me, but believe these pictures. I think pictures are worth a million words. These are just some of the patients in the study. It does work. You need to find the right patient, obviously. And I'm super excited. We are starting probably this month, or not this month, in June. And we just got IRB approval. A NIAID-funded study, large study, U01, with several collaborating centers. Some of them are here, to study it in children. We are super, super excited about this. We also are now analyzing 20 children that we treated successfully with this, that had AD as well. So with that, I hope you believe me that alopecia areata is now increasingly recognized as part of this atopic march and has type 2 inflammation in those patients with atopy. So the take-home message in general, many immune and barrier mechanisms are shared across atopic conditions, but it's important to note that there are some unique characteristics for each, and increased understanding of the common and unique processes for each one will improve our ability to develop targeted treatments and also targeted prevented strategies for the entire atopic march. With that, I want to thank you. This is my department. It took, as you imagine, a big village to do all of these studies, and I want to thank all of them, and I want to thank my funding sources. Thank you so much. And I'll be happy to take some questions. Yeah.
So we have diseases that are associated, and we have diseases that are concurrent, and we have diseases that are sequential. And when it comes to asthma and atopic dermatitis, it can be all three. So when you said you had biomarkers that were predictive, you really mean that correlated with established asthma in atopic dermatitis patients, and not yet at the point I would think where you could find a biomarker in a 2-year-old that might predict asthma development.
Yeah, that's an excellent question. Yeah, those children already had asthma. So they had established asthma. So the two biomarkers are, let's say you take skin from a child, you'll be able to agnostic if you know if the child has or you'll be able to tell if the child has or not asthma. But I can see how this can also, lead to a predictive biomarker. Yeah.
Hi, very compelling study for the asthma and the tape strips. So I wanted to ask, was there any special consideration for the location of the tape strips? Or maybe ask it a little differently, do you think you would see that effect in different areas of the skin where you sampled?
That's an excellent question. So in all our AD studies, we try, if possible, to take the tape strips from the extremities. And indeed, she asked me, where should I take the tapes? I said, you know what, let's do it similar to what we do in children with AD, because that's also where the controls that we took are. So she tried to take it from the arm. Yeah.
Great talk. In your first talk, you showed the healthy skin with asthma in children exhibit Th1 response.
Yep.
I was wondering if the children with asthma will eventually develop AD or any skin disease when they grow up.
So, we don't yet have the answer because they were collected not so long ago, and this was not designed as a longitudinal study, but maybe in a few years, if she still has a collection of these children, she will be able to know. But as of the time when the study was done, they did not have AD. And, at 7 years, usually AD is developed in majority of children. So who knows?
Yes, very interesting. I'm going to assume, well, I'm not going to assume, I'm going to ask. Based on the other questions, though, was there any interest or had you taken some genetic profile of those children in the asthma study to see if they actually have a prognostic or related factors to AD, even if they hadn't developed the disease itself?
Yeah, well, this will be interesting. We didn't do that. So, and also, I want to say that here we actually, the whole idea behind it was, can the skin be a window to what happens internally. And I can tell you that we are doing now a study at Sinai with a group in GI, in pediatric GI, and we also are finding markers in the skin that can tell you what's happening in the GI tract. And I think the idea is the skin is the largest organ in the body, and likely, of course, you need to have severe disease, because if you have mild disease, you'll not be able to detect it in the skin. But if you have more severe disease, likely you will be able to see it in the skin.
Thank you. Good morning. I have a question. In your study about tape strips and the rock children and Mennonites, did you take into account, air pollution? Because there's a lot of Korean studies correlating atopic dermatitis severity in air pollution.
Definitely, yeah. So in this study, Kirsty, that does really a Herculean work here. She and her team at Rochester, they look at all the markers, including pollution and so many things. You need to understand that the team from Rochester actually goes to the houses of these children, because you can imagine they will not come to the doctor's office. So the study is designed so that she and her team are going to them with dry ice for collection. They do all the collection there. It's a very fascinating study that I think will shed some light into some early on mechanisms that lead to atopy in general. But yes, she looked at pollution. Some of her studies are published from the first cohort. The Zoom one is published.
Such fascinating work, and I'm so interested in this eosinophilic, non-atopic alopecia areata. And I think we have pretty good hypotheses for why Th2 mechanisms develop in the skin, and I was wondering if you have a favorite hypothesis for how this eosinophilic response develops in this multifocal way in alopecia areata.
That's an excellent question. So from what we see that about half of the patients with alopecia areata will have this atopic alopecia areata. But the studies done by the DermPass community, these were agnostic. So the numbers I showed you, these are from the entire alopecia patients. So 80-something% you saw will have mast cells, and around 30% will have eosinophils. And we do see sometimes, in my own studies, we do see sometimes atopic cells, even in those not fully associated with atopy. That's why you saw that dupilumab also worked to some extent, even in the other population, but not as well. Probably it was more indirect. So maybe it took more time, and people will not wait 2 years to have the starting of their hair growth. I think in the interest of time, maybe we will move on now to have Giulio Sipovic tell us more about neuroinflammation. He is a very dear friend of mine for many years, and he comes to us from Miami, where he directs one of the biggest itch centers, and he's also the Stifel Chair of Medical Dermatology, and super excited to have him here.
Well, thank you, Emma. It's great to be here. I have an interest. These are my disclosures, and I'll today discuss, usually I discuss itch, but Emma asked me to discuss neuroinflammation, and it goes together, but the major issue is here that there are interactions of nerves, keratinocytes, and skin, and I'll try to share with you these interactions that go up to the central nervous system, and there are a lot of key players here. I won't be able to cover all of the mechanisms in AD, but it's an exciting field, and I think that the skin is also a great place to understand the neural system. So when we talk about, all of us are aware about the issue of type 2 cytokines activating itch, and we now know that we have also expression of these receptors for these cytokines in the nerves. But it's important to understand also that neuropeptides are extremely important, and some of them are such as substance P, CGRP, activate also the immune cells, whether it's eosinophils, mast cells, also macrophages, dendritic cells, and T cells to secrete these cytokines. So there's a really good crosstalk between all the system, amplifying a neurogenic inflammatory component that activates itch, and I'll discuss later on also pain, which is something that we neglected for many years in the suffering of atopic patients. So I think this is a very simplified scheme to show that cytokines and non-histaminergic pruritogens activate nerves, and downstream, and the transcription factor, JAK-STATs, and downstream there are ion channels in nerves, the TRP channels, that are extremely important also in a neurogenic inflammatory component. The most common are the ones that I'll point out. TRPV1 is more about histaminergic and is associated with substance P. But the one that in atopic eczema is always overexpressed is TRPA1. This is also the receptor for the Japanese wasabi. And TRPV3 and TRPV4. TRPV3 is extremely important actually in keratinocytes and barrier function. And these are all also overexpressed in atopic eczema. TRPM8 is interesting. It's that cooling, that's the receptor for menthol. Just to let you know that there are patients, we always tell our patients that this is more on the practical level, we always tell our patients to do cold showers, but in fact 30% of atopics would tell you that itch is aggravated with cooling. And by the way, TRPM8, interestingly, this is unrelated, but in psoriasis, actually, this receptor cooling the skin actually aggravates itch. Another important aspect is a protein that is associated with a type 2 inflammation, periostetin, and I see my colleague, Dr. Mishra, who was the one who found that periostetin, we found during a period that it was overexpressed, and we thought that this protein was only associated with type 2 inflammation. And Santosh then found that actually this periostetin is activating integrin alpha beta receptor, not just in keratinocytes, but directly in nerves. So this protein is kind of a double whammy, inducing itch and scratch and aggravating, again, associated, aggravating itch, but associated neuroinflammation in atopic eczema. Another key player, and it comes back to Santosh, is that brain natriuretic peptide is a neuropeptide secreted by DRGs. All those of you who are clinicians know that actually brain natriuretic peptide is associated with congestive heart failure. But interestingly, this neuropeptide has been reported by my colleague Dr. Steinhoff and a Chinese group associated with atopic dermatitis. I would mention that this neuropeptide is not unique just to atopic eczema, but it is a very common neuropeptide associated with itch of different types. And now I'd like to discuss a bit another G-coupled protein cell receptor, MRGPR. In particular, this is a group of proteins that are extremely important in neural transmission of different types of itch. And most of them are located in nerves, but the MRGPRX2 is unique, where it's not been yet reported in nerves. It's possible that there is some expression in nerves that we did not yet find, but it's highly expressed in mast cells and in eosinophils. And in our itch transcriptomic studies, we've shown that it's highly associated with itch and atopic dermatitis. It's not just exclusive to atopic dermatitis, but it's extremely important in the non-IgE associated activation of mast cells and neurogenic inflammation. And why do I mention it so? Because I think it is a potentially very potent target for drugs, and there are several drugs in development in this field. So hopefully you'll stay tuned for that, because to me it's one of those important factors that kind of combines a lot of the neuroinflammation associated with atopic dermatitis and other inflammatory skin conditions. Now, going into a bit of a clinical arena, a lot of our patients, when they have atopic eczema, they would tell you that the skin, and it looks healthy, it's very sensitive, whether they apply a clothing or whether there's weather changes or even issues of their sweating, and they tell you our skin becomes very itchy or very sensitive. And this is a neuronal phenomena called neurosensitization, and the definitions are allonesis or hypernesis. What is allonesis? Allonesis is a non-itchy stimuli causing itch, and that's why they put, the changes in weather or clothing induces their itch. And then hypernesis is a mild itchy stimuli causing more itch. And when we did a study way back with my colleague, pain expert, Lars Arnedet Nielsen, and his postdoc who spent time in our lab, we've shown that even a mechanical stimuli in patients with atopic eczema induces a significant reaction in healthy, in lesional skin, and even in non-lesional skin in patients with atopic eczema, and particularly also in patients when you induced a non-histaminergic pathway of cowhage. And this figure that my fellow just recently kind of produced is showing you that the mechanism here is called a neurosensitization. And the neurosensitization encompasses a lot of aspects. It's both peripherally and centrally, and it involves neuropeptides, and it involves signaling also of barrier disruption, also microbiome. Heidi Kong is here, and there's a lot of now interactions between microbiome and nerves that we know, and it goes on, but it's important to understand that the neural system is overactivated in patients with atopic eczema. And this overactivation is also reflected in activation of cytokines directly, and that's why the drugs like dupilumab, nemolizumab, the JAK stats, all the new biologics, Trelo and Lebri, they work very rapidly on itch even before you see the changes of inflammation, because there are receptors in the nerves that are blocked, whether they're IL-4, IL-13, and IL-31, and by blocking these receptors, there's a reduction of neurosensitization. And this is a study that my former fellow, Takashi Hashimoto, has done, nicely showing that blockade of by dupilumab reduced that neurosensitization quite quickly after 1 week. So clearly, you didn't see yet the changes in inflammation and eczema so significantly, but you could see that the neural system is already tuned and has a reduction of response. But on the other hand, what is interesting is that although you would think, I mean, as a clinician, a lot of times patients say, well, if you have more itch, you'll have more nerves in the skin. And in fact, this is an interesting phenomenon that we've reported almost 15 years ago, and now I think it's quite clear to the field is that the neural system is actually in the epidermis is downregulated. If you could see on the healthy, this is with a skin clearing technique. It's another method that we learned from my former fellow Tae Hong Lei from Singapore developed this method, is you could see significantly high expression of nerves in the epidermis in healthy, but in atopic dermatitis, even in non-lesional, but in particular in lesional, there's a downregulation of nerves. So you would say that's really contrary to that, all this thought process of neurosensitization. But we think that there is actually nerves that are inhibitory for itch in the skin, and they're downregulated. There's also an apoptotic effect of some of the antimicrobial protein affecting these nerves. So interestingly, and this is part of a study, different stat, that we've shown that with dupilumab, there's a significant, look at the baseline, there's almost no nerves in the epidermis, and end of treatment, there's significant expression of nerves in the epidermis of patients with atopic eczema, suggesting that the drugs can affect not just the physiology and the immune, but also the anatomy of the nerves. So another aspect of a neuroinflammatory component is the pain. And it's interesting because almost 20 years ago, we presented that a lot of patients with atopic dermatitis had pain, but dermatologists were not interested so much in pain of atopics. And later on, several years ago, Jonathan Silverberg kind of confirmed that, and we now know that pain is an important component of atopic dermatitis. About 40% of the patients have pain. And this kind of is related in a way to that neurogenic inflammatory component. And there are a lot of common features in terms of expression. So I mentioned the MRGPRs. I would mention also protein activating receptor 2. This is the receptor for serine proteases that is highly expressed in patients with atopic dermatitis. Again, a lot of the TRP channels are overexpressed. There's also sodium channels in the nerves that are associated with itch and pain, and also some other neuropeptides, endothelin, bradykinin, and serotonin, and also nerve growth factor. There are some unique features of itch and pain that are different. I mean, periostin has not been shown to be involved in pain, and we know that there is an imbalance, and Emma showed a very nice study way back that there is a imbalance between mu and kappa that could be with a drug that is a kappa opioid being alternated in patients with atopic eczema. So clearly there are a lot of similarities, but there are some differences. And if we discuss neuroinflammation, we have to also understand whether there is an effect on the brain. And I, for many years, investigated the role of brain in itch, and patients with atopic eczema is the prototype of a chronic itchy condition. Clearly, scratching is a response that the brain kind of induces. It's not just a reflex, but it's important to understand that there are a lot of psychological aspects, and they're related to neuroinflammation. And in fact, that a lot of the patients will tell you, I have an attack of atopic eczema when I'm stressed or any activity that causes stress. So clearly, it's important to understand why the brain is involved in atopic eczema. And we've shown that. This is a study that my former fellow, when he was at Wake Forest, we did Yozo Ishijo. We imaged the brain of atopic eczema and compared it to healthy subjects when we induced itch. And in fact, there are brain areas that are specifically highly associated in itch in comparison to healthy subjects. And the areas, maybe won't be surprising after I kind of told you that there is that neurosensitization. These are centers that are also involved in neurosensitization of pain, such as the anterior cingulate cortex and the dorsolateral prefrontal cortex. And in fact, there was a significant correlation with the severity of itch and also with the disease severity EASI score with activation of these areas. So again, suggesting that there is this brain-skin connection and neuroinflammation. And as I mentioned, stress is so important in these issues, and clearly some of the treatments before the era of biologics that I was using to reduce itch and atopic eczema were dealing with drugs that affect the brain, such as neuroleptics or antidepressants. And one of the studies we've done was to see whether acute stress influences atopic eczema itch intensity and also scratching. This is a test called Trier social stress test. And what you could see here is that, interestingly, the itch intensity did not increase, but the scratching was significantly higher, spontaneous scratching in atopics, after it's not so fun to stand in front of these researchers and talk, and you have to subtract numbers, and you have to talk about your achievements in life, and clearly it's stressful. And in fact, you could see that the amount of scratching is significantly higher. And we've also looked at brain architecture. Again, these are things, when I studied in med school almost 40 years ago, and we were taught that the brain is basically irreversible after being an adolescent. And we now know that there is actually brain plasticity. And what is interesting is that the hypothalamus, which is significantly associated with stress, the level of gray matter was significantly reduced in atopic eczema, and it was highly associated with the reduction, it's in inverse correlation, with the stress levels, suggesting that there is here, again, anatomical damage of some sort in patients with atopic eczema. I won't show you because we did not publish that yet, but with a German psychologist who came to work with us, we've looked at patients after they were treated for their atopic eczema and improved significantly, and in fact there's changes in gray matter. So again, that brain plasticity is something that occurs in patients with atopic eczema. So kind of to summarize is, I know that I'm a big I would say that I'm fond of biological treatments and targeted treatments, but we have to remember that, first of all, these treatments may have an effect on the brain. It's correct that these drugs don't penetrate the blood-brain barrier, most probably, but they could cause changes in the periphery that are translated into the central nervous system. But it's important for me also to explain to patients that psychological stress is something that we need to address, because it, in a different pathway, could reduce the neuroinflammation and even cytokine expression. I believe that, to conclude my talk, is that I hope that you have the same kind of feeling that there is a significant component in the peripheral nervous system and central nervous system of neuroinflammation. And this leads also to pro-inflammatory cytokine secretion in atopic eczema, and that neuropeptide and cytokines are key players in that neurosensitization in atopic dermatitis. So with that, I'd like to thank a lot of those folks that have been working with me on these issues, and all my funding agencies, and I'm open for any questions, whether it should be now, I don't know, or later. Thanks. Yes, okay.
I can start. Just amazing talk and so many complex pathways. It's really amazing how you're able to present all of that so holistically. Thank you. I was curious and actually really fascinated about, I think it's functional MRI that you had presented there, and just interested in knowing whether or not in your atopic patients versus the healthy subjects, was there more extent of activation? Were there more multiple foci of activation, or was the intensity in the specific areas? So do you see?
Yeah.
Yeah.
Excellent. I mean, there are two components here. So the brain is activated in many. So there's not just one brain center for itch. There are a lot of areas involved. And you're right that there is a lot more activity in patients. What we looked also, you have to understand in the functional MRI, what we did is we induce an itchy stimuli, and we compared also the itch intensity versus, so when we kind of take that as a cofactor, still there is significant differences between the atopic and the healthy. So it's not just, it's a correlation with itch intensity, but I'm taking that as, because you have more itch intensity with cowhage versus in healthy there's less. So it's important. What I showed also, the anatomical are totally different. This is just gray matter, so that was the component of the functional MRI. The functional MRI with voxel brain morphometry is just measuring the level of gray matter. So there is difference in gray matter between, that was surprising. It means that there's a dynamic event, and I'll explain to you where we now know in patients with depression, and we didn't look at depression at this, but patient with depression, we know that there's less gray matter. And when you treat these patients, there is, again, plasticity and regeneration of nerves. And this is something that, to me, is the next generation of understanding how things are working. And it's beyond just atopic eczema. So I think in the future, a lot of the drug companies that are working now on developing drugs, it would be interesting for them to understand. I know the drug companies are a bit afraid of dealing with drugs that, who knows, maybe it causes some kind of multi-encephalopathy, or leukodystrophic changes of these drugs, which don't happen, but, from the past. But I think it's important to know how the brain is changed and whether there are any beneficial effects in the brain. And I think our well-being of our patients is a part of it. Yes?
So, Gil, do you think that the peripheral neuroanatomy is a response to the itch, that there's a pruning of nerves in response to itch? And I guess there's nothing non-invasive in imaging, because of course we'd be really interested in looking at babies where, the changes might not be seen there and you might be able to see sequential changes.
Yeah, it's an excellent question, both of these questions. I mean, I would say the following. I'd love to be able to do studies in babies. I just don't think that it could be done. I do believe that, as we know, the itch-scratch cycle also induces that neurogenic inflammation and more neuropeptides. So it's a really interesting part looking into it. I do believe that the infants, when they don't scratch so much, but they do other activities that are, whether they can't sleep. I mean, I think that the sleep aspect is not just related to scratching. There is some component there of neural that itches, and I think we'll need to better understand how it works. My only concern, tape stripping, I did a lot of skin physiology before I did itch, worked with Howard Maybach, and I have to admit that the problem with tape stripping is, unfortunately, we can't get information about the neural system, or even skin biopsies. We're limited because, remember, a lot of genetic material is actually in the DRG. It's a bit farther away. So that's the challenge. But on the other hand, there are some signals in the periphery, too, that you could find. Yeah. Any other? Okay, thank you again. Oh, I'm sorry.
Thank you. I'm wondering if you have had any experience doing tractography in AD patients. I mean, we already know, as you show us, that there are changes in the gray matter, but have you seen if there are changes in the tractography images?
I couldn't, what?
Tractography is a new imaging that allows you to see how are the tracks connecting in the brain. The neurosurgeons are using it for preparing for surgical interventions.
Are you speaking about white matter? I can't.
Yeah,
yeah.
the tracks.
So yeah, we've tried actually to do that way back when I was in Philadelphia. It's more complex at the moment. I don't know. It's a very good question. Yeah, sure. Okay. Well, thank you again.
Thank you, Gil. Really brilliant lecture. Thank you so much. And I'm happy to introduce somebody that doesn't really need a lot of introductions, Alan Irvine, the current president of the IEC. Unfortunately, he couldn't be here in person. He sends his regards. And this will be a pre-recorded lecture on mechanisms and biomarkers of atopic dermatitis prevention. I think there is no better than him to give this lecture. So Alan Irvine, that I think all of you know, he really is a leader in studies of atopic dermatitis and also other diseases in children. He's a pediatric dermatologist in Ireland, in the Trinity College Dublin since 2007. And he is the current IEC president. And he published many publications, got many international awards, including awards at the AAD. And we are super excited to hear this lecture. So can you put this lecture on? And by the way, after the lecture, we'll have a 15 minutes break.
For joining us in the International Expert Council meeting today at the SID in Dallas. I'm really sorry I can't be there. So this is a pre-record for you, and I hope this adds to the content of the day. I'm going to talk on mechanisms and biomarkers of atopic dermatitis prevention. My name is Alan Irvine from Dublin. First of all, I'm going to talk a little bit about what we know about the epidermal barrier in AD. We're going to introduce the concept of preclinical atopic dermatitis, have a review of how biomarkers may predict development of AD, and the state of the art in barrier prevention. As a reminder, atopic dermatitis has a complex and interacting pathogenesis. There's skin barrier abnormalities, type 2 expansion, also type 17 and type 22, but predominantly type 2 expansion, Staph aureus and other microbiome abnormalities, all of which drive the cardinal symptom of itch. Let's just take a look at the barrier. And the first barrier is a physical one. That's corneocytes linked together by desmosomes with very complex lipid structures in between. The kind of classic bricks and mortar analogy where the corneocytes are bricks and the mortar is multiple complex lipids, phingolipids, ceramides, and so on. Within the keratin filaments, there are filaggrin molecules which bind these filaments together, and we'll talk a little bit more about filaggrin in later slides. Proteases allow cleaving of the stratum corneum, a falling off of the outer layer of the stratum corneum and a shedding of that. And filaggrin degradation products are important in flexibility of the stratum corneum and moisturization of the stratum corneum. This is collectively called NMF. Filaggrin is not the only source of NMF, but it is the dominant source of NMF. And in this physical barrier, there are also preformed pools of cytokines which are waiting to be released by proteases, and they're part of the innate immune defense, IL-1 alpha, IL-1R alpha, IL-1 beta. There are multiple preformed pools of these as part of innate defense mechanisms. And cytokines, apart from IL-1 and IL-1 beta, we can see, for example, IL-33, TSLP, and IL-23 are all prominent in the outer layers of the skin barrier. There is a second barrier, and that's a tight junction barrier, and that's maintained between stratum granulosum 1 and stratum granulosum 3 layers. And that's normally where Langerhans cells and other antigen-presenting cells rest pseudopods in a calm barrier state. However, when the barrier is activated and inflamed, the tight junction barrier, we know, opens up and it allows extension of pseudopods through that barrier, and it allows antigens to be presented from the external environment to antigen-presenting cells, and then the beginnings of adaptive immunity, including adaptive allergic mechanisms. In atopy and atopic dermatitis, there's a tight relationship with a disrupted skin barrier, and the question is, well, which comes first? And there's no simple answer, much like the chicken and the egg. There are individuals who have very clearly got disrupted barrier first, and then there are others who perhaps the immune system becomes disturbed first. And I'm going to try and tease that out a little bit in this talk. The evidence for a primary epithelial defect in atopy is strong from genetic skin diseases. The genodermatoses teaches this. For example, in ectodermal dysplasia, this is hypohidrotic ectodermal dysplasia, which has a strong association with skin inflammation and allergic sensitization, including food and aeroallergens, as well as other allergic diseases. And that's predominantly a structural barrier in the skin. Netherton syndrome, similarly, due to overexpression of proteases because of a lack of the SPINK5 gene, which gives a regulatory function on proteases. We mentioned proteases as being important in stripping the stratum corneum. Here, if protease activity is unbridled and not regulated through Letke, the protein that's encoded by SPINK5, we get really broken down stratum corneum too rapidly and multiple allergies, including distal allergies like eosinophilic esophagitis, food allergies, and others. Corneal desmosome, you remember in the first slide, the desmosomes binding together the corneocytes. Well, we've known for some time now that loss of function of corneal desmosome gives a peeling skin syndrome, again with systemic sensitization, allergies, and an atopic dermatitis-like pathway. And similarly, desmoglein, again, another desmosomal protein deficiency there leaves severe dermatitis, multiple allergies, and metabolic wasting, coined as SAM syndrome by Eli Sprecher and Liat Samulov when they first described this. So we have a series of these nature's lessons that we can tell from profound barrier defects driving allergic sensitization, type 2 inflammation. So we know that barriers can really drive this in extreme senses. I'm going to talk a little bit about filaggrin, and filaggrin is a green stained protein here in the stratum corneum. It consists of a huge amount of the molecular weight of the stratum corneum. Here we can see nuclei stained blue and keratin 14 stained red within the basal layer. Carrying a filaggrin loss of function mutation is around 10% of Europeans, Han Chinese, Japanese, and many other populations outside of sub-Saharan Africa do carry these mutations. It's profoundly deleterious to health. It carries a much increased risk of atopic dermatitis, of peanut allergy through sensitization through the skin, and even of asthma, but only in people who previously have had atopic dermatitis. And we've known this for some years now. So barrier defects in rare diseases cause type 2 inflammation and allergies, and similarly, barrier defects in common mutations cause this too. And of course, filaggrin is not just a driver of a causal factor in atopic derm, but it's a modifier. So if you have a filaggrin mutation and atopic derm, you're more likely to have earlier onset disease, more severe disease, cheek and hand predilections, palmar hyperlinearity, more likely to get infection with herpes simplex virus and staph, more likely to have asthma, including treatment-resistant asthma, and a more higher risk of food allergy compared to those who have atopic derm and who do not have filaggrin mutations. So filaggrin is quite a useful stratifier or dichotomizer within atopic dermatitis. And we're going to come back to that in some later slides. So first of all, I want to look at some early events then at the skin barrier. And when we're born, we move from the most profound environmental challenge that we have in our lives, really through the entirety of our lives. We move from an aqueous, xenobiotic, entirely sterile intrauterine environment to a dry environment, a xerotic environment immediately afterwards. And of course, at the same time, or almost simultaneously, we begin to acquire the microbiome. There's no microbiome in utero in the skin, but it's acquired, and it's mostly from the mother and from other people in the house. So we go from this very 100% aqueous environment to a xerotic environment and acquire microbiome, and these are very profound changes. These have been known about for up to 40 years ago. People started studying filaggrin breakdown, and this is a classic study by Harding where neonatal rats were delivered by cesarean section and then immediately had their skin occluded or not occluded. And in the occluded rats, there was no filaggrin breakdown, and in the non-occluded rats, there was very rapid filaggrin breakdown. So the skin has a way of sensing whether it's in an aqueous or a xerotic environment, and it regulates filaggrin production according to the humidity of this. There's a very extreme example from looking at those birth changes, going from birth to 100% humidity by just occlusion and birth to just normal air humidity. It has a profound influence on how the skin behaves. And how this, the exact mechanisms of this, the sensing mechanisms for this and the transcriptional control are not fully understood, but it's worth looking at a little bit at filaggrin and its post-translational processing. So obviously we have the filaggrin gene, mostly exon 3, encodes the profilaggrin, which is in keratohyalin granules. That's dephosphorylated and chopped up by SASPase into filaggrin monomers. Monomers then bind keratin filament granules, bundles in the stratum corneum. They're broken down by bleomycin hydrolase, also caspase 14 and caplin 1. And finally, we've NMF in the outer sense of the stratum corneum. So the NMF is quite a good proxy for filaggrin breakdown because it's the end result of filaggrin post-translational processing. That can be measured on tape strips, or it can be measured through Raman spectroscopy in vivo. So we're going to show a little bit of data from this. So Raman spectroscopy is a very sensitive way of picking up the amount of natural moisturizing factor in the stratum corneum. And it can be done very reliably on the thenar eminence, and it can be done right down to newborn babies. And we've done that, and we'll show some data just about what happens immediately after birth. You can put tiny little hands on one of these portable units and get really quite accurate and predictive values for whether that individual carries a filaggrin mutation or not. This is using the River D diagnostic system of Gerwin Pupils and colleagues. And here's some data that we just want to show. And in blue, you have wild type filaggrin. And what you can see here is the age in weeks. But particularly if we look at those very early days, those first few days after life, where filaggrin gets broken down extremely rapidly, just like we saw back in the 80s with the Harding experiments in cesarean sectioned rats. We can see this now in human babies having regular measurements of NMF immediately after birth. And we can see that it rapidly increases up to around 2 weeks and then begins to stabilize out towards a year. These are studies that we did in Cork, looking at babies who were born and immediately had their filaggrin mutation. We had filaggrin mutation measurement, but we also then did Raman NMF. And if you look at the filaggrin loss of function carriers, we can see they dichotomize out really early, even at 2 weeks. They're segregated very tightly. So we did full sequencing with of filaggrin through next generation sequencing with John Common when he was working in Singapore. So we have a pretty good handle on who's got a filaggrin mutation or not. And you can see they really beautifully segregate out. And those changes are almost complete in the first 2 weeks of life. And the populations have more or less completely settled into different populations. I'm going to talk now a little bit about the microbiome. This is Staph aureus, which of course forms a biofilm over the skin, not just these little dots on it. And Staph aureus is very relevant and complex relationship with atopic dermatitis. Around 70% of AD patients are colonized. It's associated with worse barrier dysfunction. It both thrives in type 2 inflammation and drives type 2 inflammation. It's associated with food allergy and atopic dermatitis, even lack of response to some drugs. And we know that Staph aureus counts can be corrected by type 2 targeted monoclonals like tralokinumab and dupilumab. That's both been shown and published. The classic study by Heidi Kong, now 12 years old, just showing that hegemony of Staph aureus in patients undergoing flares and then the recovery to a more diverse microbial communities afterwards, more like to what the baseline was in Staph aureus. So in AD, so AD patients in the baseline have much more staph than controls, but when they're flaring, staph just takes over completely. And whether that's an epiphenomenon or a driver or both, it seems that there's evidence that it's both epiphenomenon and driver of AD flares. It then resettles, and we get these more diverse communities on the right panel after the AD has been treated. Which comes first here? There's now increasing evidence that before you can see and perceive atopic dermatitis, there is cutaneous dysbiosis. This is one of our studies, but several others, Daniel Hole and others, have shown that either a paucity of non-Staphylococcus aureus staphylococcaceae or an increased colony count of Staph aureus are both there before it's possible to clinically perceive atopic dermatitis. So I'm going to talk a little bit about preclinical atopic dermatitis in a few slides as we build a picture of what's going on in the skin before infants have atopic dermatitis. Staph aureus has a very complex relationship, as I said, with the skin, and this is a normal stratum corneum scanning electron microscopy, these nice smooth tiles built up sequentially, and it looks very smooth. However, if you have filaggrin mutations, you do get very dimpled stratum corneocytes, and there's an index that we can call, we can actually quantify this as a dermal texture index depending on the number of pimples that are on the corneocytes. And what these pimples express is cornea desmosine here with an immunogold electron microscopy staining. These little pimples are on villi are decorated with lots of cornea desmosine on the surface. And the reason that's important is because Staph aureus can bind to cornea desmosine. And we're producing not just smooth bricks like on the left, but more like stickle bricks, a much greater surface area for Staph aureus to bind to, therefore much harder for Staph aureus to be cleared from the skin of patients who have these abnormal corneocytes. This is work that we did with collaborators in Belgium looking at clumping factor B and its ligand, and it turns out that the ligand is indeed cornea desmosine. So what we have here is a single live Staph aureus with a little bit of cute nanotechnology showing the strength of the binding to these ligands, these cornea desmosine ligands. And we can see that clumping factor B ligands are very strongly expressed on low NMF corneocytes. So as we go through those changes after birth, and those low NMF corneocytes are produced in filaggrin mutation carriers, their tips are decorated with cornea desmosine, and then Staph aureus finds it extremely easy to bind to those tips, and it's quite difficult to shift Staph aureus off that. And if you create a mutant Staph aureus that has no clumping factor B, it just can't bind at all, as in the right-hand panel. There's no binding, there's no decoration of these corneal villi. Of course, there's a second hit on the barrier in atopic dermatitis. We have these intrinsic structural defects and we have the microbiome defects, but there's a big hit through the activation of the type 2 immune system. So I mentioned earlier at the top of the talk, these alarmins, IL-23, IL-33, and TSLP that expand basophils and subsequently through innate lymphoid cells expand IL-13 and recruit eosinophils and mast cells into the skin. And this is a stress response to the epidermal barrier. And what it does is it causes downregulation then of filaggrin and many other epidermal barrier genes and proteins, loricrin, involucrin, filaggrin-2, and many others. So there's a vicious cycle of inflammation going on here. So what about the concept of preclinical atopic dermatitis? This is something Amy Paller, Tiffany Scharschmidt, and Sonia Kejik and I published on recently in the JID. And we think it's important because there are a number of studies exploring this phenomenon. This is from Jacob Thiessen's group in Denmark, but there have been many others. Here, his group found skin TARC and skin tape strips in predicted childhood atopic dermatitis in clinically unaffected skin in clinically normal infants at 2 months of age. And in fact, several groups in Denmark, South Korea have looked at preclinical atopic dermatitis and found TSLP, phytosphingosine, TARC, IL-13, TSLP, and complex changes in ceramides that predict and predate atopic dermatitis development. And we think this is an interesting concept because whilst not many of these studies have been reliably replicated because different groups have looked at different markers, we can see that there's patterns emerging. Here's some work from my group looking at S100A8 and 9. And what we find interesting is it's just been published in the Journal of Allergy and Clinical Immunology. What we find is that there was a stratification whether the children carried filaggrin mutations or not. So in filaggrin mutation carriers, which we can think of as being a profound structural barrier abnormality, we didn't see these inflammatory markers, S100A8 and A8, 9 dimers. They were not elevated at 2 months in patients who subsequently developed AD. But in filaggrin wild types, they were very strong predictors of 6-month AD, 12-month AD, and cumulative 6 and 12-month AD. So people who had, the children who had high levels of S100A8, 9 were much more likely to develop atopic dermatitis, but only in individuals who did not have filaggrin gene mutations. And you can put all of these studies together in a graphic just like this. And this is in this month's episode, this month's edition of the Journal of Investigative Dermatology, looking at preclinical atopic dermatitis with the co-authors I mentioned earlier, the first author of Amy. So we see in preclinical AD, there's already a lot of the machinery of AD is there, but the skin isn't itchy and it has normal appearance. It's no symptoms, but we see decreased NMF, increased reduced ceramides, high sphingomyelins, low free phytosphingosine, high TARC, high TSLP, high IL-1 alpha, IL-8, IL-18, increased S100, A8 and 9, Staph aureus colonization, and lower microbial diversity. And as that converts, and of course it doesn't convert in everyone, but they're much more likely if you have that profile to convert, then those biomarker profiles carry across, but we see the much more cellular infiltration with T cells, of course, mainly type 2 T cells, but others, eosinophils, type 22, Th17 cells. And we see the clinical appearance of erythema, vesicles, excoriation, and of course it becomes itchy. So this transition from preclinical to clinical AD is really fascinating and perhaps is a window of opportunity. And we can summarize that a little bit more easily graphically by just looking at the infant cheek, low NMF, ceramide and free base alterations, type 2 skewing, and microbiome alterations. So can barrier protection prevent AD? Well, it's a mixed picture is the answer. So this is a meta-analysis of 10 trials. You can see it's skewed by the very high numbers in 2 very large trials. And some studies show a skin intervention, going back to 2018, 2019, there are studies in smaller numbers of individuals which show protective barrier intervention from birth does lead to less children getting atopic dermatitis, but some very large studies don't. And of course, these studies are so complex and so mixed in terms of the emollients they used, the duration that they used them, when they initiated treatment. But perhaps these data give you a clue, the ones I showed you earlier, and this is the filaggrin very early window of opportunity. So you'll remember some of the biomarkers, the inflammatory biomarkers, are not so important, the S100, 8, 9 in in filaggrin mutation carriers. So what about very early intervention in the first few days of life, when all of these changes going from that aqueous to the xerotic environment, when all of those changes are active, what if you went in very early and just for a short time, for 8 weeks, until you see everything is largely settled down? So this is a study which we did and published last year. It's a single center RCT. We looked at high risk for AD, that's family history, a 1-to-1 randomization for either standard skin care or an 8-week intervention twice daily with a Veno baby daily care, baby gentle wash was used for cleansing. And we used this for just 8 weeks, and we saw that in the intervention group there was substantially fewer children developed atopic dermatitis compared to the control groups. And this is just a schematic of the overall study view, 8 weeks, and then they were followed out to 12 months. And we looked at AD at 6 months and 12 months. And we found that there was a significant difference between the including point prevalence and cumulative prevalence at 12 months. Even with only 8 weeks intervention, there was a long-lasting effect on point prevalence and cumulative incidence at 12 months. So what we then did was stratify this by filaggrin mutation status. And what we essentially found was that filaggrin mutation carriers were much more sensitive to this intervention, and the number needed to treat in filaggrin mutation carriers were much lower in numbers compared to filaggrin wild type. So this intervention was effective overall on a cohort, but we saw a much greater effect size in filaggrin mutation carriers, which again might make sense because they're the group that have the perhaps fewer inflammatory markers in the preclinical stage, but the most profound barrier defect as well. And what we can see here is that in the control group, the filaggrin loss of function mutation carriers obviously have much more atopic derm than in the wild type. But when you look at the intervention group, there's no difference between the two groups, and both are lower than the control group, substantially lower. So we begin to see that effect size playing out. What might have been a little different in our study, there was very good adherence to the study protocol, and perhaps that helped differentiate it from some other early intervention studies. So just to come back then to atopic dermatitis being a complex condition, a complex disease with multiple interacting pathways in its pathogenesis. We talked about skin barrier dysfunction, we talked about immunological skewing, and talked about the microbiome. But of course, we know that by far the most tractable pathways in this are immunological pathways, either through monoclonal antibodies, oral small molecules, or topical small molecules. So that introduces the intriguing possibility of very early intervention, either through systemic therapies or perhaps through topical therapies in at-risk individuals, either before or immediately after they first develop atopic dermatitis. And what effect would that have on the overall risk of developing AD? Those studies are not yet done, but we can see that there's some, at least some proof of concept ideas that might support that. And this is an older JAK inhibitor, tofacitinib, not licensed for atopic derm, but used in vitro. And we can see that inhibition of JAK-STAT3 will lead to improved barrier function and improved filaggrin expression, for example, as well, and improved larkin expression. So JAK inhibitors at least in vitro systems, can upgrade filaggrin expression, and that's an intriguing possibility for future interventions, as well as the possibility of topical corticosteroids, but perhaps they'll be less useful because we know that topical corticosteroids actually downregulate NMF and filaggrin expression in the round. So just to conclude, barrier dysfunction is a cardinal feature of atopic dermatitis. Preclinical AD has many changes in predictive biomarkers, but these are not widely replicated because of the nature of how the studies have been done. Those dysfunctions are driven by genetics in the microbiome and amplified by immune reactions, and they can be modified by both topical drugs and systemic therapy. And this is perhaps where barrier repair and barrier prevention is going to come down to, is knowing which individuals are most likely to benefit from which type of intervention. And we just simply haven't got the information to really be definitive about that. And it's currently poorly understood. So I want to thank lots of people who've worked with me in the data that I presented here. These guys want to thank you too for continuing to study their skin. And I want to encourage everyone to interact with the Eczema Council. Thank you for attending today. If you're not a member, please do consider joining, and please do look at our website and look at our education materials. And I hope you find it useful. So thank you for your attention today.